It appears that we are officially live. Hello, everybody, and welcome to another session of our Sussex Vision Seminar Series, as always within the Worldwide Neuro Initiative. I'm George Kafedis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from University of California, Berkeley, Professor Karthik Shekhar. Following his dual degree in chemical engineering from the Indian Institute of Technology in Bombay, Karthik went to MIT and the lab of Arup Chakraborty for his PhD entitled Dimensionality Reduction in Immunology from Viruses to Cells. He then joined the Broad Institute of MIT and Harvard as a postdoctoral fellow with Aviv Regev and collaborated in a number of visual system projects with Joshua Sainz. And then, just before the pandemic hit in 2020, he moved to Berkeley and University of California as an assistant professor in chemical and biomolecular engineering. In his lab, they use single-cell genomic approaches and statistical methods to understand the patterning of neural diversity. For that, they focus on different aspects of the visual system, from building a physiological cell atlas of the human eye to discovering cell type specific profiles of vulnerability to injury, and then to the topic of his talk today, the retinal types across different vertebrate species. I'm very excited to have Karthik here with us for his talk entitled Comparative Transcriptomics of Retinal Cell Types. So without any further ado from my side, please all welcome Professor Shekhar. Karthik, the stage is officially all yours.
Thank you, George. All right, is my screen visible?
Yes.
So thank you, George and Tom, for the invitation. I've been a big fan of this series for a long time, and therefore it's a great honor to be presenting as a speaker. So let me begin by just saying that our first vertebrate ancestors, they roamed the earth about 550 million years ago, and their diversity in terms of the number of species greatly expanded during the so-called Cambrian period. And these vertebrate species had a visual system whose components, such as different types of photoreceptors, were also inherited from earlier bilaterian animals. So there's a great evolutionary inheritance that was already there. However, over the ensuing 550 million years, the vertebrate visual system has been evolving in its own right to accommodate a diversity of visual needs, let's say from fish that inhabit shallow waters to deep sea angler fish that you see here, from amphibians, from aerial birds and predators, to species, a variety of species living in the land in different habitats. And even within the land, we have species with different visual needs, from those that operate in daylight, from crepuscular species to primarily nocturnal species. So there's a great variety of visual needs that are accommodated by the vertebrate visual system. But the remarkable thing that's been known about the vertebrate retina, which is the neural film in the eye that initiates visual processing, is that its basic blueprint has been largely conserved across all the extant vertebrate species. And by the basic blueprint, I mean the existence of three nuclear layers where all the cell bodies exist, and then two synaptic layers, which where these cells, they form synapses. And this blueprint contains 5 neuronal classes and a glial cell type that is specialized to the retina called neuroglium. And so looking further in, it's also well established for a long time now that these different cell classes contain a diversity of cell types that are really discrete entities in terms of morphology, function, and also molecular profile. And we also know that these cell types, they organize into highly specialized circuits within the retina to enable feature detection, which results in the retina's ability to communicate parallel channels of visual features to the rest of the brain. So the question naturally arises as to what extent are the cell types and the circuits conserved across vertebrate evolution, and what is varying and sort of what is constant. And so this talk is going to focus on cell types and not so much on circuits. And so classification of cell types in the retina has a very rich and venerable history, beginning all the way back to Cajal and Golgi. And then since then, we now have increasingly comprehensive atlases where the cell's identity is defined either based on morphology, based on function, light responses, or increasingly based on molecular signatures. The earliest sort of molecular work was based on single antibodies, genes, or transgenes. And I should say that, while these methods built up a great body of ground truth for later methods to come, one of the limitations is that they don't permit straightforward evolutionary comparisons, primarily because a lot of these features, it's difficult to compare them across evolutionary timescales. But ultimately, evolution is encoded in the genome. So there was a need for a genome scale readout that could both resolve cell type and be leveraged for evolutionary comparisons. And so the advent of single cell genomics or transcriptomics, which happened when I was, at least high throughput methods happened when I was a postdoc in Aviv Regev's lab, paved the way. So I'm not going to get into the details of these technologies because by now they are fairly well established, and I'm sure many in the audience are also familiar with it. But briefly speaking, these are ways to measure the transcriptomic states in single cells at very high throughput. So ultimately, what we get from these measurements, which can be droplet-based, or there are several other technologies like split pool barcoding, is that one gets a data set where thousands to tens of thousands to hundreds of thousands of cells, each of which is defined by a gene expression or an omic vector. And ultimately, these vectors can be used to decode the identities of cells, identify cell types, and also identify other biological processes through machine learning approaches. And so where this really becomes powerful in the comparative sense is that years of comparative genomic work has now provided clear correspondences of genes throughout the genome that are orthologous across species. So two orthologous genes derive from the same ancestral gene in that sense. So by analogy, we can define the notion of orthologous cell types such that gene expression patterns across these orthologous cell types, one cell type in species A, another cell type in species B, will be statistically conserved. But just as orthologous genes themselves, let's say in these two species, they ultimately accumulate mutations, orthologous cell types are also allowed to accumulate divergences in gene expression patterns over a period of time due to either evolutionary drift or natural selection and various other approaches. So there are two claims that ultimately form the foundation of this talk. The first claim is that the omic vector that you can measure from these assays, which may include the transcriptome, it could also include the epigenome or the proteome or the lipidome, several such assays are being developed. Ultimately, these vectors are stable proxies for a neuron's phenotypic identity. And that's the first claim. And of course, we know that this is not always perfectly true, but we know from a lot of work that it's largely true. And the second claim is that the omic vector of a cell type can be leveraged to infer evolutionary relationships. So I just want to briefly review some old work, which was the first foray into us testing claim one in the retina. In fact, it was my first project on the retina as a postdoc, which is focused on bipolar cells, which are these excited neurons that reside in the inner retina. And so back then in 2014, when we started this project, 2015, we had a pretty good and a pretty comprehensive census of bipolar cell types in mice, thanks to the work of Heinz Wessler, Thomas Euler, and several others. And 13 morphologically defined bipolar cell types had been found. And as you can see in the schematic, the major axis of variation in morphology is how far into the inner plexiform layer these axons of these bipolar cell types arborize to. And so this became our ground truth. And the question simply was whether each of these morphologically defined cell types had a clearly identifiable and stable transcriptional signature. And we showed in this paper that the answer is indeed yes. It turns out to be a clustering problem. And these bipolar cell types beautifully cluster out into discrete molecular clusters. And of course, to assign them specific names, we had to look at what genes are specifically expressed, and then combine this information with a sort of morphological readouts, in this case, lentiviral labeling with fluorescent in situ hybridization, to sort of match each molecular cluster to its morphology. And in this case, we were able to identify or match each of those 13 morphologically defined types to a single molecular cluster in our data. But what was also surprising is that because this was an unbiased profiling approach, we found two types that had not been described before. One was a subtype of the bipolar 5 group, which I'm not going to get into the details of. But the other was surprising because it had a bipolar cell's transcriptome, but not a bipolar cell's morphology. In fact, it looked like an amacrine cell. And so we went ahead in terms of calling it a unipolar or bamacrine or whatnot. There were several debates on what to name it, but we ended up calling it BC1b because it was sort of homologous transcriptionally to the BC1a subtype. It's a fairly boring name. And what we showed was that it is indeed a bipolar cell. It was also present in the EM data published by others a few years ago. And what we and Rachel Wong's lab, who arrived at this cell type through an independent approach, showed that it actually starts its life as a bipolar cell, but ultimately loses its apical dendrite. And Rachel Wong's group had a much more interesting name for the cell type. They called it the gloomy glutamatergic monopolar interneuron, also as an ode to the amazing weather in Seattle. So over the next few years, we sort of took this enterprise forward with many other colleagues in Josh's lab, where we defined a taxonomy for retinal ganglion cells in mice, and also more recently, Wenjun Yan and others defined a taxonomy for amacrine cells. So just to summarize, this was the mouse retinome, and a major sort of contribution of these studies was to provide molecular markers for all cell types in the retina. Of course, it's always possible that we missed a few cell types here and there, but at least studies in ganglion cells from several other groups using several other modalities have shown that the rough number of 45 ganglion cell types is correct in mice. And more recently, a work from Greg Schwartz's group, Getz et al., and another group, Huang et al., have shown that there's a pretty good correspondence between molecular signatures, morphology, and physiology for several ganglion cell types. Okay, so now comes to the meat of my talk, which is to look beyond mice and ask sort of how conserved are these cell types. And this, again, has been a study long time in the making. It was one of those studies that I sort of dreamed of when I was doing my postdoc, but only came to fruition when I had started my own lab at Berkeley. And this is a collaboration with Josh Sains. So this is where sort of we're profiling retinal cells across multiple species and hoping to use these gene expression orthologies to ask if we can systematically understand which types are present in which species and which types are evolving faster. And so I just want to acknowledge that this was a collaborative effort, but the sort of heroic work was done by a student in my lab, Josh Hahn, and a postdoc in Josh's lab, Abuzar Munawar Fashani, who carried out the computational and the experimental aspects of this work. But we have contributions from several other collaborators, including Yirong Peng and several others listed here, who worked with us on this project. And there's a preprint out a couple of months ago, which you're welcome to look at. And so just to go over the species composition, we have 17 species that include 3 primates, humans, the crab-eating macaque, and marmoset. We have 4 rodents, mouse, the deer mouse, the Paramiscus, grass mouse, and also the ground squirrel. We have 3 ungulates, the cow, pig, and sheep. And we have 3 other sort of miscellaneous mammalian species, including the tree shrew, the opossum, which is a marsupial, and a ferret, which is a carnivore. And then this is a mammalian-heavy paper, but we also have a few non-mammals from the chick, brown lizard, zebrafish, and sea lamprey. But as I'll tell you at some point, because of both the undersampling of non-mammalian vertebrates and also for technical reasons, it becomes a little difficult to actually establish clear correspondences between mammals and non-mammals, and that's the next frontier. And over the last couple of months, although it's not part of the paper, we've added a couple more species. So we have the rat, which is another rodent, and thanks to Olivier Marr, we also have data from the mouse lemur, which is a primate, although a more distal primate compared to macaques and marmosets. Okay, so first things first, in terms of just doing brief sanity checks, what you can see in this data once you do the first level of analysis, is that the same cell class is more similar across species than different cell classes are within a species. And that's sort of illustrated in these correlation heat maps, where what we're doing is we're computing the pairwise correlation of a cell class in a species with another cell class in a different species. And what you can clearly see in the left heat map is that there is this block diagonal structure where all Mueller glia across all species sort of are more similar to each other than they are to photoreceptors and other cell types. And the moment I arrange this heat map instead of by cell class, by species, you can see that these correlations disappear. And this is also much more viscerally obvious when you examine the conservation patterns of well-known class markers that have been used in rodents primarily, such as VSX2 for bipolars and OTX2 for photoreceptors and bipolars and so on. And all these genes are also quite well conserved across these species, all the way extending to lamprey. So suggesting that class-specific signatures, which are obvious by morphology and even by physiology, are also fairly obvious based on gene expression patterns. This is also true as we discussed for retinal subclasses. So for example, if you look at rods versus cones, the famous transcription factors like NRL and NR2E3, which have been known for a rod fate specification, are also seen across different vertebrate species. The same thing for on versus off bipolars, and the same thing for GABAergic versus glycinergic amicals. So just to briefly review the data, this is sort of where we are at right now. And I should briefly mention that we have not simply done a unbiased profiling of the retina, because that usually ends up in a lot of photoreceptors depending on which species you're operating in. So to enrich for the more interesting, at least in our view, cell classes, we've had to use FACS-based methods to enrich for either ganglion cells or bipolar cells, which were the two species that we have been looking at. So overall, we are at about a little over 1 million cells throughout all species. But primarily what I'm going to focus on, which has also been the subject of this paper, is the diversity within bipolar cells and also ganglion cells. So just to review sort of how the analysis proceeds, we get data from every species. And the first step is to apply a clustering pipeline to every species separately to define the diversity of cell types within that species. So for example, this is the diversity of tree shrews, where we basically identified 13 molecularly distinct bipolar cell types and 41 molecularly distinct ganglion cell types. And same thing in humans, where we find 12 bipolar clusters and 21 ganglion clusters. So this sort of analysis, which essentially involves the standard steps of single-cell RNA sequencing, sort of data crunching, from normalization, from dimensionality reduction, to sort of clustering and also visualization, is performed in every species separately. So we have atlases for every species. So the next question is, how do we start comparing these atlases across species? So we, in fact, this sort of thing we did well before this paper in a study which was led by Yirong Peng, where we were studying the cell types in the macaque fovea and the macaque peripheral retina. And so both for mice and primates, there was a good morphological atlas of bipolar cell types. And back in 2019, we created a gene expression atlas, which is shown on the right. Okay. Now, what we can do is we can use these gene expression atlases to establish correspondences across cell types. And the analysis sort of briefly proceeds as follows. What we do is we train a multi-class classifier, which could be a random forest, a neural network, or gradient boosting trees, what have you. It doesn't matter. The method does not really matter. On one of these atlases, such that given a cell and its transcriptional vector, the classifier knows to assign it to one of these 12 cell types in that species. Now, it's important to note that this classifier is not trained in any way on the other species. It's trained only on one species. And then we apply the classifier to the other species to sort of transfer labels across species. The question is, if I take all the cells from BC5A, do they map to one of the primate clusters, or do they map throughout? So that's the sort of analysis that we do. And the result is illustrated in this confusion matrix, where the rows are the primate cell types, the columns are the mouse cell type, and the size and the color of this dot is telling you sort of how many cells in this primate cluster map to a particular mouse cluster. And so the conclusion basically is that for 10 out of 11 primate cell types, we can find a clear sort of one-to-one match in a mouse cell type. And if you draw the correspondences in the sort of morphological table, you can see that the morphological numbers don't always correspond. For example, primate 1 maps to mouse 2, primate 2 maps to mouse 4, and so on. But of course, the on versus off polarity is conserved, and sort of largely things are conserved. And then if you look at, let's say, for 4 pairs of these, what I call orthologous cell types, so one pair involves a mouse cell type and a primate cell type, you can clearly see that there is a signature of conserved gene expression which ultimately underlies our ability to infer these matches using this machine learning pipeline. Okay, so that's mouse and primate. This study was about extending the sort of analysis to several other species. And so here we begin with about 13 or 14 gene expression matrices, which we now hope to integrate to infer these correspondences. We leverage the presence of these orthologous genes. So for the species that we are analyzing, we can identify approximately 6,500 one-to-one orthologs, such that every gene has a single correspondent in every other species. And the next procedure is to apply an integration pipeline to integrate all these data. And ultimately, the goal is to take data from all these species and project them onto a low-dimensional latent space where hopefully all the species are mixed. I'll say a couple of things about sort of how this is done, but suffice to say that once we project all the different species data into the low-dimensional latent space, the hope is that clusters in this latent space, they will contain cell types from different species, and the cluster itself will represent sort of an orthologous conserved cell type that is common to different species. And so we call these clusters in this latent space as orthotypes. Simply speaking, they are groups of orthologous cell types that draw from different species. Okay. We also call them OTs in the paper. So how does this work? Beginning for bipolars, where this was almost like a test case. So if you naively sort of take all this data and perform a PCA and reduce them to UMAP and cluster, the data primarily clusters by species. As you can see on this sort of left-hand side map, they don't mix at all. And then if you overlay sort of well-established class markers for on bipolar cells or off bipolar cells, you sort of see that there is a subdivision within a species, but sort of they really don't mix together. This is the result of this integration pipeline. And I just want to briefly mention that the integration pipeline is not informed in any way by known biology. The only thing that I'm telling the integration pipeline is to take data from different species and to sort of find common anchors and mix them together. But I'm in no way telling it that all on bipolar should be together and all off bipolar should be together. So it does not know any biology. It only is being asked to mix the different species as efficiently as possible. And so here's the result. So the first thing you see is that you do find structure in this integrated sort of map where different species are overlaid on top of each other. They're mixing much better than before integration. And what's more is that if you overlay markers corresponding to rod bipolar cells, they are a subset of these on bipolar cells that are sort of this lobe, which is distinct from off bipolar cells, which are sort of these clusters over here. So it's encouraging in that regard. And I just again want to emphasize that neither the knowledge of types within a species nor known biological distinctions are in any way used to guide the integration or to penalize the integration. So it's fairly agnostic in that regard. So then what we can do is we can cluster this integrated space to define groups of transcriptionally similar cell types that are conserved across evolution. And this is the clustering. And what we did now is to sort of interpret these clusters is we asked how do the various mouse cell types distribute across these clusters. So post hoc, we ask sort of these mouse cell types, which all carry labels, which are not used in the integration, how do they distribute across clusters? And the nice thing here was we saw almost a one-to-one sort of mapping. So each mouse cell type was sort of present in one cluster, with the exception of sort of one mouse, one cluster, which is sort of with a combination of bipolar cell type 8 and bipolar cell type 9. So based on this correspondence, we ended up naming these orthotypes based on the mouse cell types that they had. So we call them ortho BC3A, ortho BC1B, and so on. So this is one integration method, and for those of you who wish to know the details, this is using a canonical correlation analysis, which was implemented in the well-known single cell package known as SORAT. Turns out you can apply other integration approaches which have been developed by others, and the single cell community is fairly open that way. And so these are the results of a variational autoencoder. This is the results of integrative non-negative matrix factorization, and this is integrative PCA. And you can see that we're pretty much getting the same and consistent results across different methods, but obviously some methods like INMF do a poorer job of mixing the different species. So based on this benchmarking, we basically decided to use the CCA-based method, which could be superior for reasons that include the better engineering of the software underlying SORAT. Okay, so with this, we then looked at these orthotypes, not just in mice, but several other species. And what was encouraging to find is that there was, each orthotype contained on average about one type from every species. So it's almost like, every orthotype is a mixture of different species-specific types. This was mammals. The mapping across non-mammals was a lot poorer. So for example, we find good matches for chick. We find somewhat okay matches for lizard. But for zebrafish, especially, we don't find really good matches. It's important to note that we are talking about large evolutionary distances here. So for example, in mammals, the largest evolutionary distance is between opossum and primate, which is about 160 million years. Reptiles and birds themselves are about, I forget the exact, but it's on the order of 250 to 300 million years different. And of course, when you're talking about teleost fish like zebrafish, we are going back 430 million years. So although I'm classifying all of them as non-mammals here, they are much more evolutionarily distant than any of the mammals that we have here. So that's definitely a shortcoming. So I just want to say that, if you look at, we can now look at these orthotypes and look for certain exemplary orthotypes. So one encouraging thing was that there was an orthotype which contained the BC1b cell type in mice. And interestingly, this orthotype had contributions from all the other mammals, okay? And interestingly, also from chicken and lizard. And so the hypothesis of the prediction would be that there should be a unipolar morphological type in all these species. Maybe it continues to be bipolar in some species, I don't know, but that's the prediction, and we have markers for it. And this is a much more obvious case of rod bipolar cells, where there's again a clear rod bipolar cell orthotype, which carries rod bipolar cells from different species. But interestingly, even though we know that chicken and zebrafish have a rod bipolar cell type, for technical reasons that I mentioned and I'm happy to discuss separately, we are not able to find, map them to this particular orthotype. So the integration seems to break down in some sense beyond the mammalian boundary. And that's, again, going to be something which we have to deal with in future work. So just to say, just to highlight the zebrafish story, much more recently, this is work from Takeshi Yoshimatsu's group, where we played a small role. It turns out that what he showed was that the cell type that looks like a rod bipolar in zebrafish, in fact, is a rod bipolar. And it not only has the morphological properties of the classical rod bipolar cell type in mammals, it's using EM and various other techniques. He also showed that the downstream circuitry also appears to be conserved. In other words, this particular rod bipolar cell type, cluster 14 in zebrafish, if you look at it downstream molecular partners, there is something that looks like an A2 amacrine cell, there's something that looks like an A17 amacrine cell, suggesting that the classical rod bipolar circuitry might really be ancient, dating back to teleost fish. Interestingly, and although this is not something I'm going to discuss, we also predict the existence of a second rod bipolar cell type in zebrafish that has a different circuitry and different properties. And if you want to know more details, you should read Takeshi's paper, which will be out soon as a preprint. So the basic takeaway from bipolar cells is that what we find from this evolutionary analysis is that the number and these molecular signatures of bipolar cell types are exquisitely conserved among mammals, and to some degree also in the non-mammals. There is a higher transcriptomic divergence in non-mammals, but there are several caveats, including sort of our present inability to fully integrate them, which may be resolved either by better methods in the future or by the inclusion of more non-mammalian species that will help us to sort of, for the lack of a better word, connect the dots of the breadcrumbs. The major variation that you see across species is the differences in proportion. So one sort of well-known difference is an unsurprising difference is the proportion of rod bipolar cells versus cone bipolar cells between species that are rod dominant and cone dominant. But even among the cone bipolar cell types, there are proportional differences. Okay, so that's as far as the bipolar cell type story. And we sort of, when we started, we wanted to use the bipolar cell types as a testing ground before we proceed to the much more interesting case of retinal ganglion cells, which are the output neurons of the retina. So again, to briefly review, ganglion cells have been studied probably far more than bipolar cells. And as far as we know, there are about 40 to 45 types in adult mice, determined by morphology, physiology, and also transcriptomic profiling. And sort of more recent work from Greg Schwartz's group, where again we collaborated in a minor way, has shown that there is a good correspondence between morphology, physiology, and transcriptomics for several types. So again, back in the macaque paper, we made the first attempt to apply the same pipeline that we used for bipolar cells to find matches between macaque and mouse ganglion cells. So prima facie, there appears to be a major difference in the sort of nature of transcriptional diversity. Mice have 45 types with no type really dominating the frequency distribution, but primates have fewer, at least as far as we can tell, discrete types. And the frequency distribution is really dominated by these two large groups of cells, the so-called midget ganglion cells, which we will talk about a fair bit in the remainder of my talk, and to a lesser extent by parasol ganglion cells. And back then, when we applied the same pipeline, we could not find any clear matches for midget and parasol ganglion cells, again, sort of justifying this idea that they're probably primate specific. Just to review, these have been well described morphologically and also physiologically. And these midgets and parasols, they're the most frequent RGC types in the retinas of all the primates that we've looked at so far, at least macaques and sort of great apes and humans. Each appears to be a paramorphic pair in that it contains an on polarity type and an off polarity type. They're obviously clinically important. So if you could only save one type in glaucoma, which is an optic neuropathy that leads to the death of ganglion cells, one would probably choose the midgets because it's what we use to see most of our daily vision. And historically, parasols have been linked to mouse alpha cells, but midget cells are considered a primate innovation, so to speak. So this was time to sort of revisit this, and almost sort of, when I started off this project, I was hoping that I would again show the same sort of lack of evidence that we had in the mouse to macaque comparison to sort of buttress the claim that midgets probably are primate specific. But we had a surprise. So the first of all, when you perform the same sort of OT analysis, now including all the species, and this again is I'm only focusing on mammals here. Before integration, you see that the cells are clustering by species, and there's a lot more mixing after integration. And again, we can define these orthotypes. For ganglion cells, we could define 21 orthotypes. And although we have, unlike bipolars, there is more variation in the number of types and relative frequency, we were quite surprised to find that at least within mammals, different species have very specific mappings to these orthotypes. Again, each orthotype appears to comprise, 1 to 3 types within every species, including from primates all the way to rodents and ungulates in the other species. And the same story is for non-mammals. Here, if you see that the mapping to ganglion cell orthotypes is even weaker, for non-mammals compared to what we saw for bipolar cells. Just purely by way of sanity checking, we find a couple of orthotypes that appear to beautifully map to the intrinsically photosensitive IPRGC types that have been described now in mice and a few other species. In particular, we find one orthotype that contains the mouse M1 and M2 as their orthologs, and another type that contains what we think is the M3 and what we think is the M5 and M6. Notably, the M4 sort of is missing from these orthotypes. And that comes to the next part of my talk, which is, do these midgets and parasols, do they have orthologs in mice and other species? It turns out that they do, and the orthologs of midgets and parasols appear to be the alpha retinal ganglion cells. So these are cell types that have been first identified in cats, followed by several species. At least in mice, what this paper from George Sains and Marcus Meister's lab shows that there are 4 types of alpha retinal ganglion cells with clear sort of morphological differences. Again, they come in 2 paramorphic pairs based on either polarity, on versus off, or kinetics, sustained versus transient. So there is an on-sustained ganglion cell, an off-sustained ganglion cell, an on-transient ganglion cell, and an off-transient ganglion cell. And these 4 types, our analysis shows, are the correspondence of the 2 midget types and the 2 parasol types. So what is the correspondence? And I have a brief schematic to take you over it. So these are the 4 sort of morphological representations of the 4 primate ganglion cell types. There is the off midget cell, the on midget cell, the off parasol cell, the on parasol cell. The analysis, the orthotype analysis, sort of the correspondence is that the off and the on midget cells in primates are orthologous to the off and the on sustained alpha cells. The off and the on parasol ganglion cells in primates are the orthologs of off and the on transient alpha ganglion cells in mice. And again, this correspondence is purely based on the orthotype analysis that knows nothing about the phenotypic properties of these cell types and so on. It's purely based on transcriptional signatures. And here I'm indicating the orthotypes where these pairs feature in. So even without providing this metadata explicitly, we seem to be matching the off types with the off types and the on types with the on types. And moreover, it's well known that midget ganglion cells exhibit sustained responses and parasol ganglion cells exhibit transient responses. What was equally surprising is that several studies have identified the dominant bipolar cell types that provide the presynaptic input to each of these types. So for example, the flat midget is the dominant presynaptic partner of the off midget ganglion cell. DB3A is for off parasol ganglion cell, and same in mice. And what was surprising and unexpected is that these pairs of primate and mouse ganglion cell types that provide the dominant input for each pair of orthologous ganglion cell types also end up being in the same bipolar orthotype. Okay, so it's almost like these orthotypes are co-evolving together in pairs. So that was quite nice, but this is one method, so this is based on this transcriptional matching. There's another method, again, which relies on transcriptional data, which appears to support these correspondences. And I want to take you through this method that was due to Marcus Meister and Mu Chao. So I just want to take you through 4 slides that conceptually explain what's happening in this method, where there's a little bit more supervision involved. So ultimately, we have a large n-dimensional gene expression space, and every cell type can be thought of as a point or a cloud in this n-dimensional space. So for example, I'm using Xij to represent the gene expression vector of a type J in species I. Okay. So for example, in this gene expression space, you could have these 4 cell types here and these 4 cell types here. Again, one Xi3 is the 3rd cell type in species 1, and X23 is the 3rd cell type in species 2. And let's assume that species 1 is human and species 2 is mouse. And so without loss of generality, let's assume that the 4 cell types that I'm seeking correspondences towards are the on midget, on parasol, and off parasol. And in this large gene expression space, again, without loss of generality, they will form some sort of 4-cornered shape, okay, in humans. And suppose I propose that these 4 cell types in mice are the corresponding cell types. Like, for example, X22 is the corresponding cell type of X12, X23 is the corresponding cell type of X23, and so on. They will also form a 4-cornered shape. So one way to say that this is a good correspondence is if the 4-cornered shape has the same shape in both humans and mice. Okay, so that's the claim. The claim is that groups of these orthologous cell types, if they are truly orthologous, will have the same sort of interrelationships across different species, with the exception of being translated across the species axis. So statistically, what I'm saying is I could imagine basically fitting a uniform model. So let's say I could approximate this four-cornered representation by this four-cornered shape shaded, and the same four-cornered shape, excepting for a species translation, would also be a good approximation to the relative arrangement of the four cell types in mice. Okay, and that's the claim. And so the way this is written up into a model is that Xij is factorized into sort of a vector A, which is a constant vector, with a type-specific vector Bj, with a species-specific vector Ci. So it's factorized in this manner, and ultimately we want to identify A, B, and C in a way that minimizes the squared residual. You want your approximation or a model to best explain the data. So how good is the fit can be quantified basically by computing the variance of the data and computing the variance explained by the model, and then calculating the fraction of the total variance in the data that's explained by the model. So in this case, for example, I'm just sort of for the sake of illustration, this particular model, which is factorized in this way, assumes that you can separate out the type part of the vector and the species part of the vector, explains about 80% of the variance in the data. Alternatively, by way of illustration, you could have an arrangement like this, where I'm proposing a matching of these four cell types in mice to this four cell type, this quartet of cell types in human. And here, the best fitting model sort of may explain a very small fraction of the total variance in the data, mainly because you can see that the mouse shape is a very poor approximator of the human shape. And so based on this, I can now take any four pairs of mouse cell types and then try to match them to the on and off midget and parasol ganglion cells and ask what is the fractional variance explained, again with the hypothesis that a true orthologous quartet should form a similar shape in the mouse space than in the human space. So we began by looking at all mouse types for which we had both information for polarity and physiology. And this ended up being a small subset of the 45 types. So in Greg Schwartz's paper, we sort of identified the polarity and the kinetics. So there are sort of four columns of mouse types here. So the on-sustained would be the putative on-midget correspondence, on-parasol, off-midget, and off-parasol. And based on these candidates, you can find a number of combinations. So the total number of valid combinations that match the polarity and the kinetics would be 9 times 4 times 4 times 3, which is 432 combinations. And so we can apply this method, gauge, to all these 432 combinations and evaluate the extent to which the fraction of variance of this model is explained in each combination. And when we do that, this is a histogram of the fraction variance explained that Marcus calculated, and the y-axis is the number of mouse combinations. And it turns out that the highest fraction of variance by far are the 4 alpha RGC types, which is nominated by the OT analysis using an independent method. And here in green, I'm showing various combinations that involve 3 alpha RGC types and one other type, which also you can see are sort of present in the tail of the distribution. Now, what's more is that this method is quite efficient. And so in principle, I don't need to restrict myself to these 432 combinations where we know the polarity and kinetics. It's fairly straightforward to apply this method to all possible combinations of 4 ganglion cell types in the mouse data. And so you have 45 choose 4 times 4 factorial, which is approximately 3.5 million combination. And if you apply gauge to these methods, again, based on the fraction of variance explained, the similarity of the shapes, the combination nominated by the orthotype comes out second out of 3.5 million combinations. And the probability that this would arise by chance, if you fit a Gaussian onto this bulk, is less than 10 to the minus 6. And I also want to mention that this you might wonder what is the top combination or the first combination. The top combination had a 0.1% larger score, but it contained matches that violated polarity based on what our prior knowledge. So for example, it was matching an on type to an off type and a sustained type to a transient type and so on. So again, this is an independent method to sort of support, at least based on transcriptional similarity, the fact that we believe that the alpha RGC types are homologous or orthologous to the midgets and parasols. So just to summarize the correspondence, we don't just have orthologs in mice, but for every sort of midget and parasol type, we have orthologs in several species. And here you can see sort of the substrate for these sort of comparisons. I'm showing a few genes that are sort of that exhibit conserved expression patterns, let's say that defining the midget off orthotype or the parasol on orthotype and so on. So I want to sort of end my talk in the last 10 minutes by speculating a couple of things. So one might wonder why alpha RGCs, why did nature pick alpha RGCs to be the orthologous counterparts of midgets and parasols? Because the surprising thing is that unlike midgets and parasols, which comprise 90% of the primate RGCs, these alpha RGC types are really not numerous in mice. They are less than 5% of mouse RGCs as far as we can tell. And so I don't know the answer to this question truly, but a clue comes from the projection patterns. So it's at least well known in mice and some of the species that ganglion cells project to more than 40 midbrain regions. But at least in mice, the principal among them are the superior colliculus and the DLGN, the dorsolateral genicular nucleus. And if you look at the statistics between mice and primates, mice, almost 90% of ganglion cells, more than 90% of ganglion cells in mice project to the SC. It's the primary retinorecipient region in mice. And about 20% of those ganglion cells that project to the SC also send afferents to the DLGN. On the other hand, in primates, it's completely flipped. Almost 100% of ganglion cells in primates project to the DLGN, which is far more elaborate in primates, containing multiple layers and so on compared to mice. And less than 10%, as far as we can tell, project to the SC. And so the dorsolateral genicular nucleus is the main retinorecipient region where primate RGCs and human RGCs projected. So one can ask, is there something special about the projection patterns of alpha RGCs? There are 3 lines of evidence that suggest that they project to the DLGN more than what you would expect based on their frequency alone. So, for example, there are these anterograde tracing experiments from the Allen Institute where they traced the projection patterns of about 88 Cre lines, and these are 4 Cre lines that I'm showing here. So, for example, this is Thai1 Cre, which labels all RGCs, and there is CDH4, NATO1, KCNG4, SADB2 that label various subsets of RGCs. And if you can see, there is one that is bright, which is the KCNG4 Cre line. And it's well known in the field that this particular Cre line labels the 4, or at least 3 of the 4 subsets of alpha ganglion cell types in mice. So again, you see robust alpha-specific labeling in the DLGN. There is another paper from the Busso and the Euler group, which does retrograde injections from the DLGN to find retro-labeled RGCs, and they classify various retro-labeled RGCs based on their physiological responses by functional imaging. And there appear to be 5 ganglion cell types in the retina that are overrepresented in the DLGN, and 4 out of these 5 ganglion cell types are the alphas. Okay. And the last piece of evidence comes from Daniel Kirschensteiner's group that basically identified ipsilaterally projecting RGCs in both the DLGN and the SC that are involved in prey capture. They identified 9 RGC types based on their morphology that are sort of dominate the IPSI projection, and it turns out that the sustained alphas are among the 9 IPSI projecting RGC types. So again, there are few pieces of evidence suggesting that in the DLGN there are a few types that dominate the projection, and in mice these appear to be alphas. There might be other types too, but it's possible that during evolution, as visual cortical processing became much more prominent, as changes happened in the rest of the brain. In the retina, there was a reorganization of the ganglion cell subtypes, and there was a numerical expansion of these DLGN preferring subtypes, and in this case, these include the alphas. So that's the sort of overarching hypothesis. So I want to just conclude my talk by saying a few things. So we've used a comparative single-cell transcriptomic approach in general, which has been quite powerful in doing evolutionary influences. We've applied to the retina. And what we find is that retinal cell types are extremely well conserved among mammals and several among vertebrates. So it's also possible to hypothesize that the circuitry involving feature detections and so on are also probably truly ancient. And what we also find is that the extent of cell type variation which is surprisingly low compared to our previous sort of presumptions, increases from the outer retina to the inner retina, suggesting that evolution is sort of working to shape the retinal output. And the sort of main finding in this paper is that the primate midget and parasol cells are orthologous to mouse alpha cells, and that their numerical expansion may be related to the increased cortical processing of vision. There are still several gaps in our ability to connect the dots between mammals and non-mammals that I've briefly touched upon. That would be an interesting sort of place for future studies. And again, the big question, at least in my view, is how did cell types co-evolve across different brain regions? Because they don't evolve independently of each other. And ultimately, how can we connect this to the evolution of genes and enhancers? Because that's sort of where evolution is really happening at the molecular level. So before concluding, I briefly want to touch upon the cell class that sort of we didn't cover in this paper, and just to give you some unpublished results, is the amacrine cells, which are probably the most complex cells of class of cells in the retina. Again, they've been classified fairly well in mice, where Yan et al. Find about 60 different types of amacrine cells. And although a lot less is known about amacrine cells compared to ganglion cell subtypes, there are several amacrine cell types that have been fairly well characterized. These include the A2s and the A17s, which are part of the rod bipolar pathway. We have the catecholamine expressing amacrine cells. We have the starburst amacrine cells that are intimately involved in the direction selective circuit in the retina. And then we have the SEGs and nitric oxide expressing amacrine cells and so on. And when we perform a cross-species integration of amacrine cells, which is led by a graduate student in my group, Dario Tomasini, again, what we find is that among mammals, there's a beautiful conservation. You find clear orthotypes corresponding to SACs, corresponding to A2s, corresponding to A17s, the VGLU3, the SEGs, and the NOS, and several other orthotypes, suggesting that these even less well-characterized amacrine cell types may be conserved. And these are these confusion matrices, across 3 species. And as you can see, there is variation in primates, but at least as far as you can see for mouse and opossum, there appears to be a clear conservation of species-specific types to orthotypes. So I will end with that by acknowledging various people. So first of all, I want to acknowledge my group, Josh Hahn, who led the evolutionary work, Ayush Kumar, an undergraduate who contributed some aspects of it, and Dario Tomasini, who's now working on amacrine cells. The key collaborators, Josh Sains, who has been a great mentor and also a collaborator in this work, Abuzar, who led the experiments, Yirong Peng, and then Marcus Meister and Mu Chao, who did some of these independent statistical calculations, and several contributors who have been partners in this enterprise. And thank you for your attention, and I'm happy to take any questions.
Thank you so much, Karthik, for this fantastic and truly comparative work. There are already a lot of questions appearing in the chat, and I would like to remind to our audience that apart from this first round that I will moderate with the questions that already appear there, we will have a informal, traditional gathering in the Zoom room that we're currently using, and I already posted the link for that. And starting with the questions, the first one is from Wei. It's quite a general one, I think a question that all of us have. If I didn't learn anything about the gene analysis things, what is the first step I should do?
Oh, is this Bailey?
The YouTube name says Wei, so I guess it could be Wei Li, but I cannot confirm it.
Sorry, so the question is, if I don't know how to analyze
exactly, how would I proceed? What would be the first step?
Yeah, so I think the first thing I would do is to go on these sort of packages. So if you're using Python, the package that's popular is ScanPy, and if you're using R, the package that's popular is Surat. They have extensive tutorials, and they also have ready-made datasets for you where you can analyze data. And then what we've also done over the course of several of our papers is uploaded our datasets and also the code in Surat and R for, let's say, the bipolar cells, or even the comparative study that I've talked about. So all of this is publicly available. So I would start by just learning the ropes by doing general analyses in Surat, and then the first thing you could do is to do the retina. So in fact, any new student that joins my lab, usually the first sort of project is to take an old data set and apply new methods and sort of figure out if we were wrong somewhere. So that's the
right.
That's the first thing. And I would say compared to especially methods in physiology and morphological analyses, gene efficient analyses has a much lower barrier because there's a good culture in the community to make the methods and the code open source right away.
Right. I would say that the first step would also be contacting the experts, as is the case here. So the next question that I will ask you to address, Karthik, is from Pierre Matar. Brilliant story. One of the hypotheses explaining cell type specification is the deployment of combinatorial codes of transcription factors. I'm curious whether you think that transcription factor codes could explain the orthotypes.
Yeah, so I think, thanks for the question, Pierre. So I think we can definitely find transcription factor codes that will separate, one orthotype from the other. But there's a lot of redundancy, and it's a little difficult to sort of, based on that, to conclude without additional knowledge that these are the minimal sort of terminal selectors, as Oliver Hobart calls them, that are responsible for the specification of one orthotype or the other orthotype. In fact, I would assume that transcription factor codes may be much more conserved across orthotype than effector genes, but we've not done that analysis very carefully. And I would also say that it's possible that some of these codes may actually be present during development when the specification is happening, and then sort of go away, when that is happening. It may not be completely obvious based on their adult transcriptome. So that's the other caveat.
Thank you very much for this. Next question is from Tom Baden, and it concerns the polarity of on and off cells. So he says, from your charts, it looks like off alphas, both the transient and the sustained, are much more obviously conserved in non-mammals than the ons. Could that mean that the off is the ancestral state and ons emerged later with some sort of duplication or something?
I mean, that's a great question. We do find that there is a greater conservation of the offs, and also in some species, at least as far as I'm aware, the off numbers are much higher than the ons. As to their evolutionary origins, it's something that I don't think we can conclude any of this based on the analysis that we've done. So really, I can't say which is the ancestral type, and probably the thing to look at would be. I don't have a good answer to that. I'll have to think a little bit more on how sort of one can conclude that offs are really the ancestral state. So if that is truly the case, you will expect to find off signatures sort of much more broadly among all cell types than you would find on signatures, if something is an ancestral state. But again, I don't know how to frame that.
Right. There are a lot of questions appearing in the chat, but if I may take advantage of my moderator rights and ask one of myself. So now that we are on the polarity topic of on and off, Sampath and Fain showed that in lampreys you have, instead of on rod bipolar cell, you have off rod bipolar. And you said that it breaks down when you leave the mammalian phylogeny, but I was wondering where this off rod bipolar would be fitting the orthotype, let's say, for these rod bipolar cells.
Good question. I mean, I don't know. If we included it right now, we won't be. I mean, I would imagine that the off rod bipolar cell will be homologous to the on rod bipolar cell before anything else. Mm-hmm. But showing that is a different matter. I believe in lampreys you also have these biplexiform ganglion cells that
definitely
form, and ganglion cells also that form synapses directly with the photoreceptors. So again, all bets are off in terms of what that homology would look like. But I would assume that sort of one hypothesis is that the off rod bipolar cell was the true precursor of the mammalian and the zebrafish classical rod bipolar pathway. The other hypothesis is that the rod bipolar cell itself emerged from a core bipolar cell, and the off rod bipolar cell was just forgotten. So I think all of that will be dependent on other parts of the circuitry. And I really don't know the downstream circuitry of the off rod bipolar cell.
Right. Thank you very much for that. Next one in the chat is from Marla Feller about coevolution. So coevolution across different brain regions is interesting, but it also has to be true within a retina so that analogous bipolar cell types can find analogous RGCs during development. This must mean that all the adhesion molecules and so on are also preserved, indicating that transcription factors with downstream developmental programs are
Sorry, can you read the question because I lost you briefly?
Yes. I think also like Tom is disturbing you by typing questions here. So I will restart with Marla's question. So coevolution across different brain regions is interesting, but it also has to be true that within a retina, you have analogous bipolar cell types can find analogous RGCs during the development. So this must mean that all the adhesion molecules and so on are also preserved, indicating that transcription factors with downstream developmental programs are also preserved. Marla is here with us, so if you need further explanation, I guess she can also elaborate.
Yeah. So I got the question. Absolutely. So I think when we looked at the conserved sort of cell genes within an orthotype, it definitely includes the classical conserved molecules like sidecakes and protocadherins and cadherins and so on. So absolutely, I believe that these adhesion molecules are also going to be conserved. Especially among sort of types that have been paired for eternity.
The next one in the chat is from Rasana Somaya, and I'm so sorry if I'm mispronouncing the name. Amazing to see orthotypes between species. However, do you think these datasets can also help in explaining why functional differences exist in these species, for example, between mice and humans?
Yeah, so I think that's sort of where one will have to dig into specific types and specific circuits. So, for example, we have some evidence that on DS there is an orthotype that contains on DS ganglion cells from mice, and also the recently discovered on DS ganglion cells from Teresa Putuseri's lab. That are part of the same orthotype. But for example, we cannot find yet an orthotype that contains all the on-off direction selected ganglion cells in mice, and also a ganglion cell type in humans. So it's possible that some ganglion cell types were just simply lost during evolution. So absolutely, I think the next step, which we clearly haven't done, is to do a much more elaborate census of which functional mouse cell types have a clear sort of homologue in primates, and then probably use those molecules to target those cell types in primates to see whether there is a functional sort of preservation. But undoubtedly, purely based on the numerical, the contracted numbers of ganglion cell types in primates, I would expect that several things have been lost, or at least have been shrouded.
Right. And the last question that appears currently in the chat, and I think this would be the last one also that I will moderate before we continue offline, is again from Tom. It's very targeted, I would say. Do you see a V-glut orthotype in fish?
A V-glut amicronal orthotype?
I would assume so, judging from the time he posted the question.
So fish, unfortunately, we have, this is the talk I was having with Tom. We have, in the fish experiments, which were done in collaboration with Herwig and Yvonne Kolsch, we primarily targeted ganglion cells using a transgenic line and bipolar cells using another transgenic line. And because of the amazing transgenic lines in fish, I think we didn't collect many amicron cells. So whatever we collected are too few. They just came for the ride, and they're probably non-representative. And so we haven't been able to cluster the amicron cells well. So I don't know the answer to this question, and that's why we would like to collect more amicron cells in fish.
Right. And at this point, then, I would like to thank you once again for this fantastic talk, Karthik. And of course, thank everyone from the audience that was here for another session. So let's continue offline.
Thank you.
And we are officially offline, and I officially